All right, let's get started. Welcome, everyone. It's excited to give. It's not the first IGVF workshop. The CRISPRs are first, but at least we have something about MPRA design workshop. It's organized by the Standard and Pipelines Focus Group, MPRA Pipelines. It's maybe not the work group you think it should be in, but we figured out that a lot of people that actually do this design are also attending to our focus group meetings. So we thought it's a perfect fit to also kind of do a MPRA design workshop and talk about what are the challenges in doing an MPRA design. So we have a Google Drive. You can also find it via the wiki, the IGVF wiki, and you come to this Google Drive link, where we have got also all the slides in there later on, and also the recordings will be there. This workshop should be 2 hours. We have 4 different talks, about 15 to 20 minutes, with some discussions at the end. I think we should do specific discussions or specific to specific things from the talk, after the talk, and when you have some general things, we're going to do this at the end of the workshop. And I'm pretty sure we have some general discussions here. So what do we expect from in these 2 hours? So the idea is, we want to do an MPRA, so the general goal of MPRA design. So we want to measure, I call them targets. Mike today wrote elements in the Slack channel, but we want to generate a sequence library of some elements targets. So this might be regions or variant in the genomes. And this library should be somehow robust and maybe transferable to other cell types, maybe also other assays. But the question is basically how to select these elements or targets. And when we have those, like we have a set of variants we want to test in an MPRA, what oligos, so what sequences represents those targets best. And so in general, so I add some general background. So target selection, so first of all, of course, data mining, so looking into GWAS studies, looking into publications, what's interested in. And if you come up then with a set of variants or a set of regions, it comes up with the prioritization. So so meaning prioritization has some sequence property you're interested in, some epigenetic features like it's an open chromatin, it's regulatory or it's somewhere else. You can overlap with this GWAS signals. And also use computational models. So I hope there's some of the predictive modeling groups around here, because I think a lot of people right now is some struggling in terms of prioritization of variants. And I think here predictive modeling can really help. Yeah, and there are other, like you're interested in a specific tissue that is linked to a disease, so maybe some genes of interest you have, and so on. But I always, because I have a kind of computational background, I just want to keep in mind, if we do really a lot of selection here, then it might be that we're going to have a selection bias, and it will be later really difficult for the computational modeling if we're using really complex networks. So simple things might be better. So, and the second one is, so which oligos, so which sequence represent our targets best? And so that's something that's really challenging, because just think of, that's your variant of interest here in the middle. You want to test an MPRA. And then, the question is, do you center the variant in your oligo, but maybe you overlay it with some open chromatin peaks, and you see, okay, the chromatin peak is somewhere shifted to the left. Then you look at in transcription factors, and there are some interesting transcription factors that are shifted on the right. And also on the left side, there's a repeat where you might have problems in terms of synthesizing your oligo. So this is not trivial. And I hope maybe some people show some results of this. Also about variants, how you kind of represent variants. So the classical one will be you have a reference sequence. Sorry, there's no variant in it, so it's just a reference, and you have an alternative sequence where you put in the alternative one. But it can also be other things, like you just have alternative sequence and do later a computational model to find out what's the baseline and predict every effect. And also where to place the variant in the oligo. Should be centered or somewhere on the left, right? Do we have to expect different effects if it's shifted to the left or the right than in the center? Yeah, and also the question I think will arise, what about indels? So we now change the sequence length. What happens here? So how should we deal with that? Mm-hmm. Yeah, so let's get started with we have these the 4 talks, and first Bill will start, and yeah, I'm excited about the talk. Let's try to share again.
All right, so let's see if this works this time. Window. Let's try the PowerPoint. Oh, minimize it again. Did it go black?
No, it's perfect. Perfect. Thank you.
Great. You're seeing the first slide?
Yes.
Excellent. Oh, that's great. All right. Well, I'm glad to be here. Thanks for including me. My name is Bill. I'm going to talk about some challenges in the design of MPRA STARR-seq experiments. And I'll just start by saying I don't do the experiments. So my understanding of the experiments is from the 30,000 foot level, but I sometimes look at the data that comes out of them, and sometimes I try to help people to plan their experiments from the perspective of statistical modeling. And so I am not able to change to the next slide. There we go. Okay. So I'll start by, I don't know if everyone's familiar with both traditional MPRA and STARR-seq, and so I'll just start with that. I've only ever looked at STARR-seq data. My understanding is that the primary difference is that in traditional MPRAs, the test element was kept outside the reporter gene, and there was a barcode inserted in the reporter gene. And it's the transcription of the barcode that we then quantify through sequencing. In STARR-seq, the primary difference, as I understand it, is just that the test element, instead of a barcode, is inserted into the reporter gene. So the test element is used as a barcode. We sequence the test element and then do quantification. Okay, and then also there's some differences in terms of how often synthesis is used. I believe synthesis is used more in traditional MPRAs than STARR-seq. But the point is the topics I'll be going through in the next 19 and a half minutes, well, I think will mostly be applicable to both of these, but there'll be a few cases where I'll point out that something's more applicable to STARR-seq or traditional MPRA. And so I'll point out that these experiments can be done at various scales. So people are now doing whole genome STARR-seq, where you're pooling a group of individuals' genomes and performing random fragmentation. And at this scale, you can test millions of variants in a single experiment. But you don't have complete control over exactly what the intervals are that you're testing, because you're just shearing the DNA. And of course, you can do things like probe capture and TN5 fragmentation to enrich for things like open chromatin and so on. But that you're still looking at a very, very large space in many cases. But there are also the focused approaches where you're using amplicons, where you have control over exactly the intervals that you want to test. And then synthesis, where of course you have complete control not only over the interval you want to test, but the actual sequence content. And so I'll talk about some things that are related to most of these. But I will be focusing on allelic effects exclusively. So the idea here is that you've got a bunch of fragments. In the case of, for example, whole genome STARR-seq, you may get many fragments that overlap a variant of interest, and they may have different start and end positions. And so one approach is just to align them all together, tabulate the alleles, and then you can start to look at allelic effects. And the way we do that is we align both the DNA from the input and the RNA from the output, and then we try to quantify allele frequencies and quantify changes or differences in allele frequencies between input and output. And you can formalize this mathematically, and of course we do, but I'm not going to do that. I'm just going to refer to an allelic effect. There's some allelic effect that we want to detect, and you can do that however you want. My question that I want to focus on today is, however you're formalizing the notion of allelic effect, how can we maximize, what types of things can we do in selecting our experimental parameters of these MPRAs to maximize the statistical power that we expect to get later when we analyze the data? Okay, and so the parameters that I'm going to talk about in particular are sequencing depth, number of replicates, allele frequency in your library, fragment lengths, and what I'm going to call haplotype background, and I'll define each of these when I get to them. I just want to clarify on when I say haplotype background, I'm not talking about the entire chromosome, I'm talking about a portion of a haplotype that ends up in your insert. Okay, so if you have multiple variants and you want to test one of those variants, the question comes up as to what to do with the other variants, and I'll talk about that very briefly later. And so there are two themes that I'm going to come back to several times. And the first is that I'm going to argue that the capabilities and limitations of our analysis tools that we're going to use later after the experiment's done should ideally be taken into account when we're planning the experiment. And the reason is that sometimes those capabilities and limitations will actually influence decisions we make when we're doing the experiment. And I'm going to try to influence that in this one case, at least, where we talk about the use of pooling strategies to modify the allele frequencies. So you have allele frequencies in your population, and then you have allele frequencies when you form your plasmid pool, and they don't have to be the same. So I'll talk about that a little bit later. And secondly, I'm just going to argue that simulations are just very useful. I'm going to go through an example of using simulations to do power calculations and talk about a few of the trade-offs that were a little bit unexpected, at least for me, that resulted from that. And I think these things can be informative when you're planning experiments. So just moving right ahead, with sequencing depth and number of replicates, I'm going to talk about these together because they seem in some cases to be related, as I'll try to show. And I'm going to go through this example of this power tool that we developed a few years ago. It's not the world's greatest power tool. It's fairly rudimentary, but we found it to be useful. And the way that we did this is we started with a simulator that can simulate counts. So I'm not actually simulating fragments at this point. I'm still simulating allelic counts over a number of replicates for a number of variants. And the simulator can run at any allele frequency, effect size, sequencing depth, and number of replicates. And so the idea here is to run the simulator tens of thousands of times over many different combinations of these parameters. Run your favorite estimator that you're going to use to estimate these allelic effects. So this is a model that I'm using, but it doesn't matter what model I'm using. You would want to use your model here. Whatever estimator you're planning to use on the real data, you would use here on the simulated data. And then because we've simulated the data, we know the true effect size, we can start to compute accuracy scores for each of those data sets for whatever estimator you're using. Okay, and at that point, we can start to study the relationship between the parameters, allele frequency, effect size, sequencing depth, and so on, the relationship between those parameters and the resulting accuracy. And in particular, you can look at things like power and FDR to try to construct power curves. Okay, and so that's what we did. And then we deployed this very simple tool that some people doing these experiments have found useful. So I'll zoom in on that just a second. And really, this is just intended to be an example of a very rudimentary tool to exemplify the types of things that you can do with simulators. And so just as a quick example here, I'm asking for those variants that result in a 50% reduction. So in other words, those simulation data sets where I simulated a 50% reduction of one allele versus the other allele, I specified I wanted a sensitivity of 95%, and I want to control my FDR at this level. And so the tool then filters through all the simulation results that satisfy those filters, and then presents the results in terms of these choices between the number of replicates and total reads per variant. And so these provide choices for the experimentalists in terms of their experimental design, in terms of replicates and sequencing depth. So a couple unexpected insights, at least unexpected for me, were that there does, at least in some cases for some parameterizations, seem to be the synergistic trade-off between replicates and total sequencing depth, where larger numbers of replicates do seem to allow you to get away with lower total sequencing depth per variant, not just sequencing depth per replicate, but total per variant. Now, a caveat here is this is based on simulations. And obviously, the accuracy of your power calculations will depend on the accuracy of your simulation. But that being said, just seeing these general trends, I think, is informative. And secondly, we noticed that rare variants can require extremely large sequencing depths. And that actually was a very important sanity check. And as I'll show, that actually prompted us to change the way we do some of the experiments in some of the labs here at Duke. And in particular, I'll describe this pooling strategy that we're using. In terms of the value of replicates, I don't have time to get into it, but in our 2020 paper, we talk about the idea that once you have replicates in an allelic assay, if there's heterogeneity in the allele frequencies between your replicates, technically this induces something called a Poisson binomial distribution, and there are interesting properties of this that we actually talk about a little bit in the paper. So I don't have time to talk about it, but if anyone's interested, there's the citation. But I want to move on now in the interest of time and talk about allele frequencies, which I alluded to just a minute ago, the idea that rare variants can be problematic. So this is going to be applicable mostly in the case of pooled samples. I talked about the whole genome STARR-seq where you're pooling a number of individuals' genomes together. So when you do that, and then here's a concrete example where a few years ago we were looking at 760 patients, you can end up with an allele frequency distribution that looks like this. And I think this is typical when you have populations of individuals that you have large numbers of rare variants. And obviously synthesis is better here because you can control the allele frequencies. But if you're going to be doing something like this with this type of distribution, it's important to be aware of how difficult it is to estimate effect sizes for rare variants when your read depths are not very large. And I believe the issue here, and I'll talk about what I'm showing here in a second, I think the issue here is that when you have low read depths, in some sense, you have reduced dynamic range in terms of the allele frequencies you can represent. And so for rare variants, with any effect size, you're talking about going from one low frequency to another low frequency. And so that is what I think is going on here. But what I'm showing, if you look just at the blue RSE curves, and these are RSE curves based on trying to classify simulated regulatory versus simulated non-functional variants. And the blue curves are where I simulated an effect size of 1/2. That means the one allele results in half as much expression of the reporter gene as another allele. And you can see that when you go from an allele, minor allele frequency of 0.05 to 0.01 to 0.005, if you look at the blue RSE curve, it really decreases in area. The ASC goes from 0.98 down to 0.72. For weaker effects, if you look at the red, in the red, theta equals 0.9, if you can read that, means a 10% reduction. That's a pretty weak effect size, at least in terms of what we can do with that. And you can see the red pretty much just follows the diagonal. But the point is that even simulating 10,000 reads, as our allele frequency got lower, it became very, very difficult to do to discriminate regulatory from non-regulatory variants. And so when we saw that, and I think this is going to underscore the value of these types of simulation-based analyses, when we saw that, we realized we needed to do something. And so people in Tim Reddy's lab, and here are some of the people who are doing this work now, realize that rather than just pulling together the entire patient cohort that we have, you can imagine that with 800 diploids, for example, you're going to get a minimum allele frequency of 1/1600, right, because they're diploids. If you instead form pools of, say, 5 individuals, now you get 5 diploids, and so the minimum allele frequency is 1/10. A nice trick. I wish I could take credit for it, but other people thought of this. And so, we know from our power analyses that now actually you have some hope of detecting, even moderate effect sizes. And so these experiments are going on now. We don't have the results yet. There are other things to figure out, such as, if you do this independently in these pools and you estimate effect sizes across these pools, how then do you combine information across that? And that's something that we're going to be working on soon. But I want to move on now in the interest of time, because I don't know how much time I have left, but I'm just going to plow ahead. Some other things that we've been thinking about lately are fragment lengths. And I'll talk specifically in the case of whole genome STARR-seq, where I mentioned that there is some ability, from what I understand, to tune the distribution somewhat, but it is a distribution. And so, just more concretely here, this is an actual fragment length distribution in a whole genome STARR-seq experiment from a pool of 5 individuals. And I want to thank Kerry Strauss from the Reddy Lab for sharing this unpublished data with us today. This is one replicate from Kerry's data, and you can see there's a lot of mass in this fragment length distribution from about, say, 300 to 500 bases. And that makes sense if you're going to be using short read sequencing, where you want to read out as much of the fragment as you get. And so to be more concrete in these hypothetical examples over on the right, for a 300 base pair fragment, if you had paired N150s, you would be reading out the entire sequence. But obviously that's a distribution, so you're going to get some longer fragments and you're going to get some unsequenced regions in the middle. Now, I don't know for sure whether this is a problem, and I'd be interested in hearing people's perspectives on this. I'm going to bring up something that may be an issue, and I'll talk about that in just a second, and I'd love to get some feedback on that. First, I do want to mention that there is some evidence now that shorter fragments may result in weaker activation in STARR-seq. So hopefully you can see that if you look at the blue distribution of activation values, or actually RNA abundance values, blue versus red versus black versus red, you can see that there does seem to be a progression in activation being stronger for the longer fragments. And this is corroborated by this very nice paper from 2020, where those authors were actually synthesizing increasing lengths. And their conclusion was that it does seem that it may be safer to go with longer fragments. And so that made me wonder about the issue of shorter fragments. If shorter fragments can have lower activation, say perhaps because they're too short to contain all of the machinery necessary to drive expression over basal levels, in that case, will that reduce our power to detect allelic effects? And that is one worry that I have. And that is why I'm wondering whether indeed it may be better to err on the side of pushing for longer fragments. What I wondered was whether there is an issue with the fact that there could be unobserved variance in the middle of fragments. Now, this is a hypothetical case. You have one fragment. You're reading the ends. You're not reading the middle. There are going to be other fragments in this whole genome STARR-seq experiment that do overlap that, of course. But currently, we're throwing these fragments away. And so one question I have is, well, 2 questions. First, do we have to throw them away? So could we do something like imputation, which I'll talk about on the next slide. But secondly, is there any downside to using long fragments? And I can imagine that if they got extremely long, you could have issues with the actual experiment, with the stability of the plasmid and the RNA and so on, or even just integration. But in terms of these unobserved variants, Certainly, I think there are cases where we could do imputation from the known genotypes if you've genotyped your samples ahead of time and you have accurate phasing. And so, for example, in this hypothetical case, whenever you see an A here, you know, because you've sequenced you've done the genotyping, you have phasing, you know that when you have an A here, you have a T here. Okay, but there are going to be many ambiguous cases when you're looking at larger pools. And I do think that there could be approaches that do this statistically. The question is that worthwhile doing? But I do think that in general, this is another case where simulation can answer a lot of these questions. So if you have phased genotypes, we can simulate fragments, we can align them, we can see how often you have variants that are in the middle that are not observed, and we can ask, is that actually a problem? What does that do to your power to detect allelic effects? If you do the imputation versus don't do the imputation, we can actually look how often do you do the imputation and so on, because we're simulating fragments. And so then I'm going to finish up very quickly now, just by talking very briefly about some questions that have come up within the last few weeks on an experiment in one lab, where we want to synthesize inserts, and the question came up as to what should we use as what I'm going to call haplotype background. And I want to be clear that haplotype background here, what I mean by that is actually just the portion of a haplotype that's within the insert that you're going to synthesize. Okay, so if your insert has multiple variants in it, then the question comes up, how do we set the alleles in those other variants if there's just one variant you're interested in, but there were other variants in that space? Okay, sorry. So just very briefly here, if you have a variant of interest with two alleles, say T and C, an obvious approach is to just put the variant in the center, and then take the reference genome and just substitute in the various alleles. You could have more than two alleles and synthesize that. The question I have is that a concern when the variant of interest comes from an association study that is relevant to some particular ancestry? And in that case, HG38 may not be fully representative of that ancestry. Again, if there are multiple variants in this region, you might want to look at what alleles are present in that ancestry in that region. And so one thing that we've considered is looking at the most common haplotype within that ancestry and doing your substitution within that haplotype, what I'm going to call haplotype background. You know, a slightly more involved option would be to instead ask things like, what is the most common haplotype containing the reference allele? What is the most common haplotype containing the alternate allele? And at this point, I think it seems to me you're really actually testing for haplotype effects. And when I say haplotype, again, I mean just the portion of the haplotype in the insert that you're synthesizing. Whereas here you're asking what is the effect of one allelic change within a fixed haplotype background. And I'd be interested in hearing whether other people have confronted this before, because I don't have the answers here, and I'm interested in hearing opinions. Also, there's, of course, the option of just synthesizing all the haplotypes within your window. And recently, I went through the exercise of looking at 250 base pair ATAC peaks and 1,000 genomes using 1,000 genomes haplotypes, and found that occasionally you do have a very large number of haplotypes for one window, but very often you have a very small number. And so that's encouraging, you know, if you're interested in synthesizing all possible haplotypes or even just ranking them. I think with a certain number, you should be able to account for a large amount of variation. Okay, and so I'm going to finish up with my last slide by saying that another question that we've had is the question of whether to put, and Max brought this up before, whether you should put the variant in the center of the insert or to one side. And it seems to me that there's a lot of prior information out there, such as annotated binding sites and so on. But I'm also wondering, and I'd be interested in hearing people's opinions on this, you know, whether there's a role for predictive modeling. In terms of choosing things like the optimal fragment positioning, I mean, there are tools out there like DeepSTARR that try to predict enhancer activity. And I wondered if tools like this or tools, other tools, could be used to prioritize the fragment positioning. And the idea here would be, for example, to choose the fragment positioning that maximizes the predicted expression of the unperturbed sequence so that we maximize our dynamic range to identify decreases in expression for those alleles that decrease. Now, also there's a case of increasing expression, so you'd want to do it differently for that. And so I'm just going to stop here and summarize by saying that I think there are multiple opportunities to create some new tools that could be useful to people designing these experiments. And I'm interested in knowing how many people have thought about using predictive modeling, how many people are using predictive modeling. I know some people after me are going to be talking about prioritizing variants, and I'm looking forward to hearing about that. But also, you know, doing things like optimizing insert lengths and positions and choosing haplotype backgrounds and so on. And so I guess I had more questions than answers here, but, you know, maybe that'll spark some discussion. And I want to thank all of you for listening to me, and I want to acknowledge a number of people that have given a lot of feedback to me from Duke, particularly Tim Reddy, Graham Johnson, and Carrie. And I want to thank Graham and Carrie for sharing their data with us today. And then also Mary Chiafani, Andrew Allen, and Eric Barrera. Okay, I hope that was no more than 20 minutes.
Yeah, thank you, Bill. It was a great start.
Should I stop the show again?
It's okay. You can just keep it.
Okay.
So I think it makes really clear that the simulation aspects before your pool data makes really sense before you're starting is your experiment, especially, and that you really show that of course low allele frequencies, it's hard to catch, or it's really exploding the number of reads you need to detect them. Yeah, we have a few time for questions. Jesse wrote something in the chat that he saw the opposite. So longer RNA fragments may have lower activity, about 2 folds for every 100 base pairs. So you can maybe read it by yourself.
Actually, can't see this here. The chat. That's
okay. You can see it. Okay, I can read the question.
Okay.
So we have seen the opposite in our experiments. Longer RNA fragments systematically have lower activity, about 2-fold for every 100 base pair, which we thought was because of cDNA amplification bias. I wonder if we need to adjust the RT or the PCR attention times to what was presented here.
Yeah, that's interesting. In terms of the experimental steps, that's something that probably other people here could comment on better than I could.
Any people want to comment on that? So same for me. I'm not on the experimental side. Yeah.
That's okay. I can just leave that in the chat, and if anybody has opinions, I'd love to hear about it.
Okay.
Another related question. I was wondering, you know, what you've seen in terms of if you have many fragments tiling the same peak or something like that. You know, how do we best estimate the strength of that peak in the genome? And in our experience, the exact sequence or fragment that you use can have really different activities depending, you know, different tiles across the same sequence can have extremely different activities. Yeah,
definitely. That's an excellent point. And I mean, there are a handful of models out there that actually do try to look at. I'm talking in terms of the allelic effects, treating the fragment position separately as an additional variable to try to control for that positioning. That's something we're starting to work on right now. So I don't actually have any real insight into that really, other than that I do think it's important. I think Jason Ernst's group has several tools that do that. Sharper MPRA, and then there's a follow-on tool to that. Is that what you're talking about, Jesse?
Yep, thanks.
Okay, yeah, great. Yeah, thanks for that. Okay.
There's a question. Hey, June, you want to ask something?
Yeah. Just actually adding to what Jesse has raised up, I'm wondering what's the definition of the long reads versus short reads. I'm wondering whether the long reads are like greater than 700 base pair or something like that, or whether it's just like 100 versus 200 base pair. I just want to get the overall range.
Yeah, no, that's a question for Jesse.
That's for, I think you basically brought up like Nature method paper or whatever, right? And from there, how did they define?
So I think, I don't remember the exact numbers in that paper, and it's worth a read. It's a good paper. I think they looked at roughly 200, slightly less than 200, and then maybe in the 400-ish range, and then maybe 600 or 800-ish range. I don't think they were synthesizing extremely large, and they had to go through some special steps to actually do synthesis that long.
I see. Yeah, I was wondering that whenever the larger fragment is there, it is more likely that that fragment will contain at least one enhancer. So I thought that it's kind of natural to see some effects like that. But I also agree with Jesse that PCR usually disfavors, right? Even the sequencing actually really disfavor this long read fragments to sequence. So yeah, I'm not sure how that was also well controlled.
Yeah. I don't remember those details from that particular paper.
All right, thanks.
How do I unshare this now? Can you unshare?
It doesn't matter when the next people are going to share, then it's
okay.
It's again. Yeah, so also great that you brought it up that about modeling the sequence, which one we should select best. So because next one is Jon, and Jon basically did already the design for the IGVF, and maybe he has some insights here, how to select the best sequence.
Well, I'm eager to hear people's thoughts. I don't know about best design, but it's where we are right now, and looking forward to some discussion around it. Can everyone hear me okay, despite the mask?
Perfect.
Great. And my screen is in regular view.
Yes.
Hello, everyone. Happy Bastille Day for those who are celebrating. My name is Jon Rosen. I am a trainee working with Haejong Won, Karen Mulkey, and Mike Love, two of which are on the call right now, and working on the MPRA that UNC is doing for IGVF. And want today to talk to you a little bit about moving from selecting variants to test and generating the MPRA sequences around those. So to start, I just want to briefly go over our initial study design. And so we are doing an in vivo MPRA study, and this would be a traditional MPRA, to use the term Bill introduced. And we are looking at allele-specific effects for 250 variants. And this is fully synthetic. So our sequences are going to be synthesized by us, and they're going to be 250 base pair sequences. Specifically, we are looking to assess tissue-specific regulatory effects by analyzing multiple tissues in the organism, in mouse in this case. We are looking to assess cis-biased effects by looking at both males and females, and also to assess gene-environment interactions by comparing allelic effects with and without perturbation. And in this case, the perturbation is going to be inflammation induced by lipopolysaccharide injection. And so we, as a group, decided to prioritize testing comprehensive sets of variants and GWAS signals. And the main motivation for us doing this really was to assist with the predictive modeling data, specifically to provide unbiased experimental results for the predictive modeling group. And because this is a concept that's going to kind of come up a couple of times, I just wanted to take a moment to clarify a little bit of terminology here. So on the left, I have a locus zoom plot. And just in case anyone here is unfamiliar with these, very quickly, each point on the plot represents a GWAS variant. The y-axis is a negative log 10 p-value. So the higher the point, the more GWAS significant it is. And the x-axis just represents the genomic coordinates with some gene labels on the bottom here. And so if we restrict our attention to what we define as a locus, so a locus is just some defined region of the genome. In this case, it's a 1 megabase region that's defined by these dashed lines. We see the lead variant, so that's the variant with the smallest p-value here is up top. And then the variants that are, we color code variants based on how correlated they are with the lead variant. And so we use squared Pearson correlation as a measure of this correlation, which we call linkage disequilibrium. Right? And so these orange and red colored variants are very highly correlated with the lead variant. And this is a common pattern we see. And so what we're going to call the lead variant and all its associated LD buddies, we're going to just call that one signal. And if we look over here, it looks like we've got some very low p-value variants over here, but we see that this dark blue coloration means that they're pretty much independent of this lead variant. And so this is going to be its own separate signal. So when we were talking about comprehensive variants and GWAS signals, what we're talking about is the lead and all its associated buddies. And the goal is really to keep them all together and maximize the number of units that we're going to analyze. And just to expand on this point a little bit more, the last locus zoom plot we had very, very narrowly defined signals, but oftentimes in GWAS results, we see something like this, where we have a lead variant and then we have a big broad signal. So we have lots of highly correlated variants with our lead variant. And so we would call this a large signal, right? Large here meaning that there's a large number of variants associated with this specific GWAS signal. And so again, we really want to maximize the number of comprehensive signals that we're going to analyze. And so the strategy we employ to do that is we take all of our identified signals and we rank them in order of increasing signal size. And so that's what this plot on the right here is showing. And so, you know, we have a number of variants that we're going to test in each round. And so in this case, it's 50,000. And if we took all of the variants in all of our GWAS signals, we're well over that. So we need a way of paring down that number, but in a way that sort of maximizes the number of comprehensive signals that we retain. And so what we do is, based on our target number, we can set a threshold such that everything to the left of the threshold, right, we just keep as comprehensive signals. So we see that the vast majority, the number of signals we keep are comprehensive. And for these very, very large signals over here on the right, rather than keeping the whole signal, what we do is we select only annotated variants. And we're going to discuss in a few slides what methods of annotation we use to do that, right? And so again, so the vast majority of our signals are going to be comprehensive. And then we're going to have a small subset of signals that are sort of biased in the sense that we're only selecting variants from those signals that are annotated. Okay, so with all that sort of preamble out of the way, we can talk about some details of our specific method. And so here's a little outline, and this talk isn't going to be incredibly technical, but obviously this process is a little bit steppy. Fortunately, a lot of these steps are pretty simple. And so we start off with a variant selection. So how do we find those lead variants that we talked about? How do we build those into these comprehensive signals? What do we use for annotation? We do a little bit of housekeeping here. How do we design our sequences around these variants? This 6 is a bit of a technical issue that we came into, and this is a topic of conversation we would like input on from everyone, and then some nomenclature, and then adding some positive and negative controls to our data sample. Okay, so let's start with variant selection. So as I mentioned before, our goal is to look at 250,000 variants, and we're going to do this over basically 5 rounds, and so each one is about 50,000 variants. And so for our first batch that we're preparing now, we are focusing on liver-related traits. And so to find these lead signals, these GWAS lead signals, we basically selected them from very, very recent, like in the last year literature. Most of these references actually come from Nature publications and represent studies that are multi-ancestry, representing very, very large studies or large meta-analyses of GWAS data. And so we're focusing on 2 main classes of traits. So lipid-related traits, specifically LDL and HDL, with a couple of other cholesterol-related traits, and liver enzymes. And these 3 liver enzymes represent biomarkers where elevated plasma levels of these liver enzymes are indicators of liver damage, specifically non-alcoholic liver fatty disease. And so with these leads in hand, now we have the problem of how do we go about building a signal. And so the first step to do is to assign these lead variants to their individual signals. And so we can't just assign each lead to its own unique signal for 2 reasons. One, a lot of the variants are actually replicated in different studies, right? So they're not unique. 2, even ones that aren't replicated, we may have a case where 2 different studies identify 2 slightly different lead variants, but when you look at the LD between the 2, they're very, very, very high. They're in close genomic proximity with very, very high LD. And so we assume that those are actually part of the same signal. And so the first thing we do is we just establish an order in which we want to add signals based on study and based on phenotype. And so we just start adding signals based on this specified order. And then if we try to add a variant in, we test to make sure that it is not in high LD or identical to a variant that's already in the study. And if it isn't, it gets assigned its own signal. And if it is, then it gets assigned as an LD buddy, let's say, to a signal that's already in there. So once we create our signal list, then we have to build out the comprehensive signal, right? And so we add in variants based on their LD. And so we identify LD buddies with the lead variants would have an LD, a squared Pearson correlation of greater than 0.8. Most of these studies are European centric. And so for the LD panel, typically we use a European panel, primarily a UK biobank, 100,000 unrelated Europeans. But we do have some ancestry-specific variants in there, specifically East Asian and African-specific ancestries. And for those, we use the appropriate reference panel for determining LD. Specifically, we use TopMed for East Asian and African LD. And then basically we add the LD buddies into signals via the same sort of order of addition that we did for the lead variants. And then at this point, we are left with just under 4,000 signals, which are comprised of almost 100,000 variants. And again, our target here is 50,000. So next step is almost all these GWAS variants are supplied reference to HD19, but as a consortium, IGVF has agreed to use HD38, at least starting out. We wanted to reference all our variants to a shared resource. We chose dbSNP, specifically build 155. And so instead of using the UCSC liftover tool, we actually did a sort of a two-step process utilizing the dbSNP file. So based on the HD19 data, we identified an RSID for a SNP. And then using that RSID and the corresponding HD38 dbSNP file, we then mapped it to the HD38 position. And an added benefit of doing it this way actually is that we could confirm that the allele identities were consistent between in the liftover between the two builds. There's additional details on this step. The uploaded files have the additional detail, but in the interest of time, I don't really want to talk about that right now. And so, as I alluded to before, we do utilize annotation, one for paring down those large signals. And so that's this first bullet point, this first enumerated point. The second thing we do is for subsequent sequence design, and we're going to talk about that in a couple of slides. But before I do, I just want to talk about what we use for annotation. So basically, there's three main categories here. One is a set of ataxic peaks identified in the multi-lab by Kevin Kern. And this is ataxic from 138 liver tissue samples. The second point is, or the second source of annotation are candidate CREs from ENCODE. And so we downloaded the latest version of this, so this version 3 from the Screen website. And Jill Moore has talked about this a couple of times, I think, in the consortium, and actually just talked about this 2 days ago in the catalog working group. And the third level of annotation actually comes from a collaboration with the cardio-metabolic focus group. So Rich Sherwood had supplied us with 9 genes that they're particularly interested in related to LDL and HDL. And so any variant that falls within 200 kb of these 9 genes, we are also annotating as well. So in my clock is disappeared from my screen, I think. Just go through this really quickly. So we just have a little bit of housekeeping, as I said before during the outline. We're just going to exclude some variants, specifically variants that fall within the ENCODE exclusion regions, right? So these are variants supplied that have a low mappability or high signal regions. Include signals if the variant is annotated via the ensemble VEP. It's identified as a stop gain or frame shift variant. And we are analyzing indels, a fair number of indels in our study. And we are accepting indels up to size 9 nucleotides. So we have a small number of identified either GWAS leads or LD buddies that are larger than that we exclude from our analysis. So in order to facilitate, at this point, we want to sort of, one, visualize the elements that we have, not just for our own benefit, but to share within other people in our group so that they can look at loci of particular interest to them and see if variants that they're interested in are well represented. We also plan on sharing this with the rest of the consortium, this genome browser track. And I just want to just sort of highlight some of the elements here. So what we have here on this first row is an individual liver ataxic peak. And what is below it actually is we build consensus peaks, and then we reduce the sort of continuous set of peaks into discrete sort of one-dimensional ranges, right? And so these are the regions that we are going to annotate with in our analysis. Down here are the ENCODE CCREs, and you can see actually some nice overlap here. On this band, we have variants that we have included in our study at this point. And so each of these little vertical lines represents a variant, and there's some identification data to the left of it. And then above that are the actual sequences, so the 250 base pair sequences that we build around each variant. And as we've already talked about, as both Max and Bill have already talked about, right, this decision of where does the variant go in the sequence. And so here and here, you see that the variant is roughly centered in the sequence. And if we look at this variant and its associated sequences, we see that it is shifted. And so that's a good segue into our shifting paradigm. So again, our sequence length is 250 base pair. And so if a variant isn't close to any of our annotation signals, or specifically the attack-seek peaks or the encode CCREs, right, it's just going to get centered in the 250 base pair sequence. And so what we do with indels, we're going to talk about in an additional slide. It's a little more complicated for long indels, right, because as has been alluded to, typically when you're comparing variants, right, you want the exact same you want the same sequence background, right, differing perhaps only on the variant. But when you have indels, right, you're changing the length of the sequence, and so that does change the surrounding environment in the sequence. But for variants that lie within or very, very close to attack peaks or CCREs, we want to shift to maximize overlap with that annotation. And so based on previous experience in our group, we have found that testing variants in their regulatory context, when you know what that is more likely to show allelic effects, right? So we have this variant 1, again, as I highlighted, right, it is pretty much right in the center of this attack peak. And so centering it in its sequence is no problem. But variant 2 here is sort of all the way here at the tail, right? And so if we're expecting that the regulatory machinery is really located more downstream from this, right? So our decision there is to shift the sequence such that it overlaps as much as possible with that regulatory element. However, we noticed that if we wanted to shift the sequence such that it really overlapped with the meat of the peak, right, the variant would no longer be in the sequence. And so that's obviously unacceptable. And so in these cases where the variant might be too far away from the peak summit to center around it, what we do is we shift the sequence basically as far as we're comfortable with, right, to make sure that the variant still stays within the sequence and doesn't get too close to the edge of the sequence. And right now our paradigm is to keep it within 25 base pair of the end of the sequence. So our pipeline utilizes 4 separate restriction enzymes. An issue that we ran into is, so we have 250 base pair sequences. These restriction enzymes have recognition sites that are 6 base pair motifs. And we ended up, 11% of our sequences contained a restriction enzyme recognition site, right? So those sequences then become unusable. And it turns out, though, that not all restriction enzymes are sort of hitting our sequences equally. In fact, 1 of the 4 is responsible for 50% of the loss due to the restriction enzyme recognition site overlap. And so the first thought is just, well, let's just replace XPA1 with a restriction enzyme that is far less deleterious to our sequence design. And so we found one that was suitable. And then the next thing we did, again, this is all motivated by, we're really trying to keep as much signal comprehensive as possible, right, for the goal of predictive modeling. And so we really did want to try to work to minimize this loss. And so the other strategy here is modification of a single nucleotide in that 6 base pair recognition site, right, so that it is basically rescuing that sequence from restriction enzyme being consumed by it. And so what we did was we said, okay, if the recognition site is more than a certain distance away from the variant that we're testing, we're okay with changing a single allele to recover it. And so in doing so, we reduced the loss from 11% to 1.6%. And so at this point, just nearing the end here. So here's just a simple case of how we build a sequence, right? So we have, I'm sorry, this nomenclature scheme is a little bit complicated. We developed this basically to be a human readable label that encapsulated all of the information in the sequence we're designing, right? So basically you should be able to take this label and and completely reproduce the 250 base pair sequence that we're generating. Right? And so hopefully this is intuitive to some extent, right? Chromosome, the beginning and end positions of the actual sequence, where in the sequence our variant lies. This is a bit of a redundant field. So this is the position within the sequence, so between 1 and 250, right? So this number here is determinable. You can determine it from the allele position and where the start of the sequence is. The ref and alt allele, whether the ref or alt is designated in the sequence, RSID, and then this is if we change an allele for that restriction enzyme site rescue plan, right? So here we have the reference allele is T, and so here's our T right in the middle of our sequence. So this is an unshifted sequence. This is the alt sequence, so there's our C. And this GGT ACC is actually the restriction, this is a KPN recognition sequence, I think. And so if we change that A to a T, right, which is what's designated here, the 186 positions change from A to T, then we recover the sequence. And so, as I alluded to before, indels get a little bit trickier, right, because we're actually changing the length of the sequence. And so our strategy is, which I believe is going to be in contrast to Nick's coming up next, is we're keeping our sequence at 250 base pair, but instead of having just ref and alt, right, instead of 2 sequences per variant, we're going to have 4 for these longer indels, right? So the first one, this is a 4 base pair insertion, right, AGAA, right, and so the ref should be intuitive based on the naming scheme. And so the first sequence we have is we're doing the insertion and we're centering this basically where the ref allele was. And then the additional 2 sequences we're adding is, so we're going to basically left justify the insertion sequence to where the C is. And so in doing so, I can't click on this without advancing, sorry. So everything upstream of this C here should match everything upstream of this. So to the left, the exact same sequence should persist. And then we do a right justified version, right? So that everything downstream of this A should match everything downstream of this C, right? And so the right side of the sequence matches. And in doing so, hopefully we capture the sort of the permutations or the combinations to align these sequences. So I'm very much running out of time. So the last thing is basically just adding positive and negative controls. Positive controls we have requested from the consortium, basically groups to offer variant level or element level controls. And as you can see here, this is what we have now, and we are still, just a call out to any groups that are here listening, still looking for additional input here, right? But already we see we have additions from Jesse's lab, from Gary Hahn, the Hitu's lab, from Mike, from Max's lab, from Martin Kirscher. This set of variants was actually suggested from the cardiometabolic focus group. Negative controls, just random sequences that avoid these restriction enzyme recognition sites. We have added some additional sequences. I just, in the interest of time, just talk about this one real quick. Again, from Rich Sherwood and Guillaume, we have coordinated with them. Again, we talked about the 9 genes of interest, and we have ensured that we have several hundred variants in our MPRA study that are actually overlapping with their CRISPR analysis. They had an additional set of variants that they identified by fine mapping that were not included in our analysis and requested that we put them in ours, and we happily did. And if other groups have variants that they are particularly interested in and would like us to test in this batch or in future batches, again, there's 4 more coming up, please let us know. Running out of time here, so I won't really harp on this, but we are also doing some haplotype testing. If 2 variants from different signals, so this is sort of a key point, right? These are not just LD buddies that happen to be near each other. These are variants from different signals that happen to be close in one-dimensional space. We are testing the haplotype combinations of those. Also, just a quick point about the shifting paradigm. Those variants, those 9 gene loci from Rich Sherwood that we had annotated with, variants in those loci that are shifted, we are also including the unshifted versions. So there'll be an internal control too, just so that we can actually quantify the effect of the shifting paradigm. In summary, lead variants from GWAS, we maintain this emphasis on these comprehensive signals to facilitate the predictive modeling groups. We implement a shifting paradigm, which we've described, and again, a call out to everyone else. Please, we are very much open to collaboration about, sharing variants, sequences, etc. And with that, I'd like to acknowledge the PIs and the rest of the group here. And I realize running low. Happy to take any questions, but obviously, if anyone has anything that we don't have time for now, I'd love to have conversations via Slack or whenever. Thanks.
Thank you, John. Thanks for the great talk, and also thank you for your huge effort to collect a lot of controls from all the others. I think, yeah. So that's really nice, and also, and you see, it's connection to Bill's talk. You thought about haplotypes, and that's also really interesting that you thought about this and put this into the essay, and it's we're looking forward for your thought. So it's okay. We're gonna have 2 questions, short, quick questions, and then we can jump to the next speaker. Otherwise, if anyone raises their hand. Otherwise, yeah, we can talk at the end. I think there will be time. Okay.
John, I have a quick question. Yeah. Just to tie it up, like Bill's talk before and your nice talk. Like, one thing that I was curious about is, like, if you guys were considering kind of like matching the background haplotype in any way when you're constructing your fragments, or is that something kind of like falls outside of the scope of what you guys want to do?
So we haven't talked about that, but actually listening to Bill talk about it, it is a very interesting idea. We have the luxury, being fully synthetic, right? We have the luxury of being able to just synthesize whatever haplotypes we want, right? We're not at the mercy of allele frequency, natural allele frequency.
Right.
But no, I mean, right now we haven't discussed any plans of doing that. But this is a dynamic project, and this is batch 1 of 5, right? And so we are certainly, even changes that we can't implement in this batch, which we're hoping to get out the door very soon. We are certainly open to considering for future batches.
Gotcha. Thanks.
Do we have the haplotypes basically because you're collecting GWAS signals?
Yeah, I mean, sorry. My question was more on the line of, like, if you're gonna, I mean, I guess you're reproducing, like, you want to be as faithful as possible to, like, possible haplotypes that you will find in populations, right? So I guess that's why the background, we call it background, I think, like, it's not exactly the combination of variants that you're testing, but also kind of like in conjunction what you normally see in the population. And I guess that there might be an effect of that is, yeah, I mean, I don't know if that makes sense, but it's kind of like another layer of, like, combination variants or haplotype or things that you normally inherit together and trying to.
I will say this. So the fact that we're using, when we're doing our haplotyping, it's from different signals, sort of forces them to not be in high LD, right? So at least we're helping ourselves out in terms of that, where we would expect to see at least 3 or all 4 of the haplotype versions represented. When if we were just taking signals that were close to each other that were in high LD, right, you would expect that there's only one predominant haplotype of the 4, in which case you're sort of testing a bunch of unnatural sequences. So it'll be interesting to see sort of what comes out of that. There's been some work in that front. I think there was it the Montgomery paper of, was it 2021, looked at the haplotype effects, and I think in that paper they saw largely they were just additive, very, very small percentage of super additive effect.
All right, thanks. I just mentioned that because just want to go over to Nick, and because Nick actually knows the exact sequence because he's so studying about 10,000 patients from and looking into de novo variants. So and in theory, he can then really put in the sequence of the patient at the position of the variant, or just using the reference. So Nick, can you try to share your screen, or should I share it?
Yeah, let me try. I think I should be able to. I had to like restart my Mac. Okay,
Yeah,
can everyone see my presentation?
that looks great. Yeah, that looks great.
Okay, awesome.
And just full screen and then, yeah, perfect.
Okay, so hi everyone, super nice to meet you. My name's Nick. I'm a PhD student in the Octave and Sanders labs at UCSF, and also part of the MPRA design focus group. And so what we've really been interested in lately is MPRA design for de novo variant data sets, which are cool and have some different properties from some of the GWAS variants from Jon's presentation. So I'm just going to talk briefly about some of our ideas regarding variant selection and design, and then also things that we've been thinking about for oligo design and for selecting control sequences. So where we sort of started for this MPRA is these type family and trio studies of autism spectrum disorder, where we have both whole genomes from both mother and father, and then from cases and their control siblings. So we're able to get a very like strong analysis of de novo mutations identified in cases and controls. So variants that these individuals with disease have that are absent in their parents. And we find about 65 of these de novo mutations per individual in our data set. And so once we did this for our entire cohort of cases and controls, we had identified over almost 730,000 de novo mutations. And this is work by Shandong, a really awesome postdoc in the Sanders lab. And where we sort of started with our analysis was taking this list of de novo mutations and sort of filtering to non-coding regions of the genome that we think might be functional. So first we removed masked regions of the genome, and then also coding regions and splice sites, since those are areas where it's much easier to predict the function of de novo mutations, and also are predictive to function differently than the sort of MPRA reporter design that we're using. And that left us with about 711,000 de novos, which is still far too many to test in a single MPRA. So what we did is we went about looking at different groups of annotation categories that we might be able to intersect with our list of de novo mutations, and then also gene lists of candidate genes that these de novo mutations might disrupt the regulation of. So for gene lists, we looked at fetal brain expressed genes and also known neurodevelopmental disorder genes from whole exome sequencing. And then we compared a few other categories, such as conservation with phyllo-P scores above a certain cutoff, functional annotations such as open chromatin regions, attack-seq peaks, CHIP-seq peaks, and transcription factor footprints within the developing brain. And then finally, we used the sequence-based model called Informer that's able to predict variant effects in DNase, CHIP-seq, and CAGE data. And we sort of used all of these as potential features to filter variants for our MPRA. So when we look at the global overlap of all of our annotation categories in the bottom, with each one of our with our entire list of 711,000 de novo mutations, we find almost 28% of these de novo variants fall within a region of suspected function, which is really cool. And then you could see there are some sets of variants that fall under multiple overlapping categories of suggested function. So those are variants that we think might be more important. And then also a large number of variants that are just picked up by more general categories, such as conserved regions of the genome with our FastCon scores, and also regions of the genome that we suspect are constrained. And then some of our other largest categories for filtering these de novo mutations, including the promoters of protein coding genes, which could be very easily assigned to the candidate genes that they regulate. And then further down the list, also open chromatin regions, and for example, are variants that are predicted to decrease gene expression from our deep learning sequence-based models. So again, this is really cool, but if we're looking at almost 200,000 potential variants, that would be 400,000 probe sequences, which is far too many that we're able to test in our iPSC neuron model. And here's just a zoomed in graph from the previous slide where you could again see these largest annotation categories pick up conservation, and then the number of overlaps between different annotation categories are smaller. And here, in case everyone wants to see, we just list all of these out. So we'll have we'll have predicted regulatory elements, transcription factor footprints, phylop as all things that we filter by. So the next thing that we considered doing is if there were ways to link these different de novo mutations to the genes that they regulate, because this could be another approach that we use to filter the number of de novo mutations that we test based on which ones are most likely to be disease relevant. So we begin by assigning de novo mutations in a hierarchical approach where we first assign de novo mutations to genes that they fall in the promoter regions of, which captures maybe about a quarter to a third of our de novo mutation data set, a quarter to a fifth of our de novo mutation list, followed by assigning de novo mutations based on contact data, and then finally assigning de novo mutations to genes based on distance or global genome architecture, such as which de novos fall within the same TAD boundaries as a set of genes. And you could see this hierarchical approach assigns almost all the candidate de novo mutations to test in our data set to a subset of genes. And this will, again, be exciting for both filtering and for like post hoc testing of which variants and categories of variants might be most important for autism spectrum disorder. And so sort of the final design that we've been thinking of right now is filtering some of these larger categories such as FASTCon scores and constraint, and then also removing the exons and promoter regions of non-coding and pseudogenes in the genome, which would leave us with a data set of about 87,000 variants to test, or 175,000 cis-regulatory sequences, which is much more reasonable size that we could fit across maybe 2 or 3 libraries to test in iPSC-derived neurons. And one of the reasons we've decided to go with as broad number of inclusion criteria as possible is we really don't have a good idea yet of which de novo variants and which regions of the genome will be functional. So we've sort of prioritized for these de novo MPRA study designs, sort of testing a broad set of variants rather than focusing more specifically. And then, so as I mentioned, the filtering approaches that we use, just to summarize, are removing certain annotation categories, filtering by certain conservation score cutoffs, such as phylop, and then also filtering de novo variants by candidate gene list, which if we needed to make this library size smaller, I think the next most logical thing to filter for similar projects would be to take a subset of genes that you of coding genes that you think are already associated with your disorder or phenotype of interest, and restrict to examples of the promoters or open chromatin regions that are predicted to act as enhancers for that gene list. Cool. So what we the next thing that we've sort of considered for our MPRA is how to handle different types of de novo variation for our oligo design. So a brief overview: our oligo will have about a cis270 base pair cis-regulatory sequence in the center, surrounded by 15 base pair adapters, which is the largest size that we could currently synthesize with at a large square from Twist Biosciences. And then the two other mutation classes that we have occurring frequently in our data sets are insertions and deletions. And in contrast to Jon's approach, where they sort of shift the CRS that they're testing left or right, we are considering using an approach where we shorten the oligo that we're testing. And our logic sort of behind this is perhaps there are regulatory regions just beyond the border of the reporter assay that are important for gene expression. And since we're really interested in a close case control study of one variant of the reference versus alternate allele, this reporter design ensures that we don't have any other differences in the reporter besides the de novo mutation of interest. And although I know some people have brought up concerns about like probe length and sequencing errors in cloning the MPRA, we're not too concerned of that at the moment because most of our insertions and deletions are short, less than 10 base pairs. So we think and hope that most of those cloning or sequencing related differences would be have a minor effect on our outcomes. And then the other thing that we've touched on today that we've been grappling with is dealing with family background. So as I mentioned in the beginning, it's cool because we have the whole genomes of both the mother and father for every child that we have a de novo mutation list for. So for example, there might be places in the genome of the mother and father where they have the child has a de novo mutation that is proximal within the same 270 base pair region to a SNP that's inherited from one or either parent. So that leaves us with a pretty challenging question of whether or not we should sort of impute all of these variants on the HG38 reference, or whether or not we should include these these family background variants. And so just a quick estimate, we'd guess that about 25 to 30% of de novos are expected to fall within the same 270 base pair window as another variant in the same family. So although we would definitely be interested in testing these nearby variants, that increases our already massive library size by another 25 to 30%. So I think what we're leaning towards right now is only including the proximal family variants in our design and ignoring distal variants more towards the edge of the CRS. And I think for de novo mutations, that makes a lot of sense. So at least we know in the coding genome that most de novo mutations there are a subset of de novo mutations that have a large effect in either like protein truncating variants or missense variants that damage protein function or eliminate gene expression. And we imagine that the non-coding mutations that are functional will probably have larger effects also and be more independent of background because of those large effects. So we think that restricting to just the proximal de novo mutations to our variants of interest will probably be the best way to balance these family background issues with our limited library size. And then the last thing that I just want to touch on is some of the thoughts in the Ahativ lab about selecting positive and negative control sequences. So I think the gold standard positive controls would be sequences already tested by MPRA in your modeler cell type of interest, which unfortunately, and I think a lot of groups are running into the similar problem we don't have yet, because a lot of groups are just starting to put out these MPRA experiments. So the second thing that we've proposed for selecting good positive controls are using the promoter regions of highly expressed genes in your cell types or model organism, because those are regions that are known to drive gene expression. and RNA-seq data is much more widely available for a lot of different cell lines and tissues. And finally, cross-tissue predicted enhancers from ENCODE. So, for example, a candidate cis regulatory element that has H3K27 acetylation activity across many, many different tissues might be a good sequence to include as a positive control if you're not really sure where else to start. And then as far as selecting negative control sequences, again, ones that are already validated in your cell type are probably the gold standard. And then one other important set of positive negative controls that we always include in our libraries are at least 100 randomly shuffled sequences from across our library. So we'll take a set of variants that we want to test and their CRSs, and then we'll just scramble all the letters in that CRS. So we know that the probe sequence is already matched for, let's say, GC content, and include that as a set of negative controls. And finally, a set of regions that are negative across all tissues is a good set of negative controls also. And then we just want to stress that including this set of negative controls is essential for proper statistical analysis at the end of an MPRA experiment, because we use the distribution of RNA to DNA barcode expression of our negative controls to estimate a cutoff of which of our test sequences are true positives. So groups should be very aware and that they include a good or well thought out set of these negative controls. And we also want to encourage for any groups doing de novo MPRA type experiments to also include control de novo variants from cases, which at the end of their experiment will help enable like interesting statistical analyses. For example, testing the activities of different categories between cases and control patients, and for discovery of non-coding regions associated with your trait of interest. So with that, I'm happy to take any questions. And I just also want to thank my coworkers in the Sanders Lab, and then some of my colleagues in the MPRA Design Working Group, and also all the members of the Ahitiv Lab, especially Tenyu Dang, who's helped mentor a lot of my MPRA experiments in the lab. Okay.
Thank you, Nick. A lot of things to test also for you.
Yeah.
Mike.
Hi, thanks so much, Nick. Can you talk about for the negative controls? Can you talk about negative control for a variant effect? Because like I agree that it's nice to be able to use the negative controls to have like a empirical idea of where you dip into the noise.
Yeah.
But then like I could see like the difference between, let's say I have a sequence where a protein does bind and there's a variant, and so that's a variant effect, and you might want to know like what is a null variant within a region where we do believe there's protein binding. What is the distribution of a null variant effect versus a shuffled sequence, the null variant effect where there's potentially no protein binding. So like, yeah, can you talk about the design of negative control sequences with respect to like shuffled sequence or otherwise?
Yeah, so that's something we're still thinking about. There are some variants that we've tested in MPRAs in the same cell type that we're waiting for sequencing results for now. So we're thinking of including some of those variants that we see don't show reporter differences in this MPRA once it's ready. But it's really difficult to get a good answer to that question. I think with these sorts of de novo MPRA designs, you benefit from including a wide variety of different variants as possible. So we have, let's say, variants that are in attack-seek peaks and conserved, that are in attack-seek peaks and not conserved, that are not conserved and or that are conserved and not in ATAC-seq peaks. And I think there's a pretty reasonable logic going in of which categories will be have more activity than others or show stronger differences. So in that sense, you could do a lot of analysis level controls that your results make sense. But definitely, like if there's a CRS where the reference has no activity and the alternate has very low activity also, that's almost impossible to draw clear conclusions on.
Thanks.
I just thought about in your case, because I know that you have also in your cohort unaffected siblings, and they have also de novo mutations. So you can also add a lot of de novo mutations from unaffected siblings, but as kind of negative control. But I think it's hard to tell because you're looking in your affected control set. What do you expect? You don't expect every variant to have an effect. It's maybe every 100 variant or 200, 400 variant has an effect. So it's hard to say that all the others from these unaffected siblings have no effect and compare them to the others. So like as a negative control set.
Yeah, definitely. And that's, I think, one benefit of going into these analyses, trying to link variants to candidate genes that they regulate. So although we might expect the controls and the siblings to have the same total number of large effect variants, we would expect different proportions of large effect variants in, let's say, the promoters of high confidence autism risk genes.
But I think you have to test a lot of these other de novo variants then to really compare those.
Yeah.
Which again, you are limited in the number of sites. Okay. Any further questions? So otherwise, it's now on me for the last talk. So here we are. Okay. So now I introduce myself. I'm Max Schubach. I'm the postdoc in the computational genome biology lab from Martin Kircher, so here in Berlin, in Germany. And I'm gonna talk about variant MPRA design that I did 2 to 3 years ago, but unfortunately I do not have the results yet. But it's I think a totally different approach than most of the others going to use, because we're a computational group, so we want to try to train models to predict variant effects. And so we thought about, so we did the KG challenge, or we provided the data for the KG challenge with a saturation mutagenesis MPRA, where others should predict the effects of these different elements. But the problem is really when you try to train a model that most of the MPRAs that are out there is just kind of standing variations, and they're mostly not rare and de novo. So EQTLs are tested. Then there are a lot of regional bias. So like, okay, these genes are interesting, or like the saturation mutagenesis, this specific third promoter is interesting, and we look at all variants, but it's not kind of an average over the whole genome. And the last problem is that they're mostly done in one cell type or cell line, and or sometimes two, and we cannot see the differences between the tissue. And it's hard to train a model. So our idea is, first of all, these are all good benchmark datasets for models, but for training, it's hard to use. So then our idea was to generate some optimal training datasets. So the same variant should be tested in different tissues. Then the variant should be somehow randomly distributed over the whole genome. The problem is not every random picked variant in the genome is fall into regulatory regions, and of course we cannot test a lot of variants, so we can test only a few. So we, in this case, in this study, we limit ourselves to this proximal promoter region. So proximal means I excluded the core promoter, and I just looked to open chromatin upstream of the transcriptional start site. And second problem is, again, if we then select any variant in those promoter, we expect no effect. Because also when we, for example, on our saturation mutagenesis MPRA data, we see about 50% of average have a significant effect. So the question is which variant we should select for testing in our MPRA. And here we're going to try to use machine learning to select variants. And that we think that might be interesting. Just before about the size of the MPRA, what we did. So so first of all, these proximal promoter regions is that we looked at upstream of the TSS, so up to 20 kb upstream, and we try to avoid, as I said, the core promoter. And because of we used a 200 base pair design for the MPRA, so we looked at open chromatin regions from ENCODE DNA tracks and just from the peak center plus and minus 100 base pairs. These are the cell lines that we want to test. So in HEC, HEPG2 and KFX is the most common, and then WTC11 cell lines. The problem is at WTC11 now there is ATAC-seq data. At this time there wasn't. We used other cells to predict on those tissues, and database that I use, and that's basically the amount. So we got a lot of proximal promoter regions. Only a few of them are open in all cell lines. Most are open in just one cell line. Quick information about the learner, how we then pick our variants. So this is also, we learned on open chromatin, so on DNA sequence, it's a convolutional neural network. We removed some chromosomes for validation in test sets, and that's the neural network. It's a convolutional neural network. It's really a really simple structure. Don't want to get into detail, but we want to really keep it simple because we want to learn something out of it, and we want to kind of backtrack it. So right now, Nick mentioned Informa, that was not out there yet, but Informa is a really complex structure, and we want to really keep it simple in terms of that we can learn something out of it. We use different strategies, so in terms of model, but the main thing what I want to say, we benchmarked our model on the saturation mutagenesis data on these elements that were tested in our cell lines of interest, HEPG2, KF5C2, and HEC 293 T cells, and these are these elements. We selected like the multitask model, so we predicted every output at once for every cell type, and that's an example. So here on the top, these are all significant effects that we got out from the saturation mutagenesis MPRA of the SORT1 enhancer, and that's our down there. That's our prediction, and you can see this for example, this significant effect we model as these one we model pretty well. Here we have more problems, but maybe we don't get the correct effect. Absolute effect size. Sometimes it flips, but in general, if we do a correlation, it's not bad, to be honest, in terms of predicting an element. So the idea was we kind of did an in silico prediction on all of these proximal promoter regions, and we selecting then positive, so active, repressive, predictive, active, predictive, repressive, and neutral effects out of them. And meaning predicted on all of those tissues, like consensus predictions, all of them predict them in the same direction with the same similar effect size, or also that are just tissue specific, so that we try to cover all of those things that we're interested in. And then I looked into transversions effects, because we also know that transversions have significantly larger absolute effect size than transitions. So the same for coding sequences, then frequent transitional the changes there, the evolution is more robust to it. So we also see this, and we don't want to have a kind of bias towards transversions in terms of high activity. So what I did is I just tried to select the k-mer content from a distribution and matched it to a background distribution of the variance. And that's just ordering the number of k-mers, and that's the frequency of it. And that's my background distribution of the whole genome. And that's the k-mers from the variants that I have in here. And then I tried to sample it after sampling. To be honest, it looks not that nice. And also I show you kind of a negative result, because so looking at the transition dispersion ratio, high effect, you see it's below 0.5, neutral effects above, and we want to get there like 0.5, but after sampling it, to be honest, it did the opposite way. It wasn't, so in total of all variants, it went better, but yeah, it did not do what I wanted to have in my data. But anyway, I just want to tell you that transversions, you have to think of, maybe you're going to expect higher effects of transitions than transversions. Next thing, and that's also, I think, really important here in terms of where to position the variant. That's the prediction. So here, this is my 200 base pair fragment. We inserted the variants randomly between 50 and 150 base pairs. So all these, they were selected between, the in silico mutagenesis was done in this range. And I just then plot the frequency of the red one is the high effect variance and neutral effects. And you see that the model predicts overall more higher effects in the center of my sequence than on the edges. And I think, I'm not sure, but I think that this will be the same on MPRAs, that on the variant on the edge have a lower effect size than if you just put it into the center. It depends, of course, around a bit where's the transcription factors and where's the core regulations, but on average over the whole genome, it might be that a variant in the center has, in theory, a higher effect than on the edge. So I try to sample that every position is evenly there. It works really well for the neutral effects because I have a lot of variants to select. It did not work. It worked also for the high effect ones, but on the edges, I didn't have so much variants to select. So that's why we have a bit of a different distribution here. But in average, it's like this. It's better than, of course, than before. Okay. Then finally, I looked into homopolymers and repeats. So that's the problem with synthesis of these oligos. And for example, I went to the CSC annotation, and there's a UCC track. And I kind of said, okay, the maximum fraction of this 200 base pair sequence of these simple repeats should be a quarter. All other sequence I just removed from my set. Also, in terms of polymer length, I took 8 as the maximum. When it's getting longer, then I remove the sequence too. So that's basically the fractions that I got to remove. And totally, I removed about 5% with this cutoffs, mostly due to homopolymers, a few to simple repeats. Control sequences. I uploaded nearly all of them also in this project design wiki folder. I used 2 variants as control sets. First, the saturation mutagenesis variants of the experiments done in the same cell types that I want to do the experiment. I used all variants with a significant effect. So have a low p-value, so significant different from 0, and at least minimum number of 10 barcodes to be sure that the effect remission might be good. And I add for every of those elements 100 random non-significant variants for each element. And so there's maybe a negative control here. And yeah, then there is this SURE SNP publication from Bas van Stensel, and it's also the citation link here. They did a kind of a whole genome MPRA in K5C and HepG2 cells, and I included the top 500 variants in both cell types. So the top significant variants as a positive control. Yeah, for the regions, because HepG2 is used, so there is a liver enhancer MPRA, so I select this, the control sequence. I think that's the same that Nick used, and also the same that Jon uses, but also I selected high active and neutral, as a neutral liver enhancer sequence from this study. Also from this study, I selected the control sequences, and finally, and that's similar a bit to Nick, is that I looked for opid chromatin and selected regions that are active in or open in all cell types present, and yeah, and active in blood and brain, so that were unrelated. So that should be the negative sequence, negative regions that are unrelated into my sequences, to my tissues that I wanted to test. So yeah, so that's again selecting using ENCODE data to select positive and negative regions. So totally, it's a large library. It's about 244,000 oligos from Agilent. So it's the length is a little bit shorter than using twist. It's 230 base pairs. So we have 200 base pair fragment and the 15 base pair adapters at the side. So it's restriction enzyme free approach. So we don't have to care about restriction enzymes. Yeah, and totally we gonna test 170,000 variants from 60,000 regions. So you see there, so I have to only, so some variants are tested. So some regions have multiple variants. So that's why I can use more of them. And finally, we have control sequences, so about 70,000 oligos. Okay, unfortunately, I don't have the results, but I want to acknowledge all the people that help here, and basically Martin and the people from Jay and Nadav's lab, and hopefully I get the data soon that I can present some data here. I think it's interesting for the rest. All right, thank you. Are there any questions? There was a chat question. Ah, okay. Thanks, Ben. Yeah, it's too late for the design, but yes, I will. I'm going to retrain the model on the DNA WTC11 when I got the results back from the lab, so that I can make an updated version of my model.
It's only a year old or something like that. Like it was
yeah.
No, I just wanted people to know that they're.
Yeah, that's great. Yeah, thanks. Yeah, and I think this somehow steps into the area so using, and a lot of publications are out that uses their MPRA data to train a model, and then say, okay, we can predict the regions here, but why not using those models to again predict the sequence you want to test, or also to know where to shift, what's the best way to where to put the variant in, and yeah, I think that's might be something that someone should consider here.
Max, maybe I missed it, but how many different promoters and variants did you test before like filtering the ones that you wanted to use for MPRA, or that you tested with your model, I mean?
I did the in silico mutagenesis on, I think, 170,000 regions. Then it's 100 base pairs where I changed to all types of variants. I have a lot of variants, but I just ranked them by effect. So I just take the top. So I don't just rank them by the predicted effect of my model. And then I had just a rank, and I just selected them from top to whatever I have space on my design.
Okay. I'm curious how computationally intensive that is. So one of the problems that we've had using other deep learning models for our genetic analysis is it's really difficult to calculate the expected number of de novo mutations of large effect within a region. And we would sort of need, in theory, like the predicted effect of every base in the genome to get like perfect estimate of that, which I don't know what sort of did you see with your analysis?
Yeah, so there are models like more complex models like some former that take a while to predict variants, and it's okay to test them in 1,000 or 5,000 or 10,000 variants, but if you test it on 1 million, 14 million, 15 millions, it can take a while, several weeks. Or you have a big TPU structure from Google in the back, then it's maybe faster. But yeah, that's a problem. That's why also we use just kind of a simple model that is faster. Maybe not that accurate, but that's not the point here. We want to learn something. We just want to kind of prioritize, and I hope that the prioritization in general is good, even if I don't expect the best correlations compared to other models then.
Okay.
Of course, now with the 170,000 variants, we definitely can test them with other more complex models, and this is okay to run in there, to run Informer on those. That's absolutely fine. But on the initial set, that would be challenging, definitely.
Thank you.
Any further questions from me? Yeah, go ahead.
Oh, if there's time.
Yeah, we have 15 minutes.
Yeah, I really enjoyed the last couple of talks, and you know, thinking towards the next round when you actually have these data sets, they of course would be really useful for building, you know, new or better deep learning models to predict variant function. But each of the data sets is designed, you know, in a special way for your specific biological question. And if we want to then sort of reuse that data set for a different purpose, like, you know, training a model to predict the effect of a variant on enhancer activity, we'll have to think. I'm curious about your thoughts about how to, you know, optimally use or combine these data sets that have been designed for slightly with different selection criteria. So for example, Max, in your talk, you know, you mentioned several times you're rebalancing the data set in various ways based on various selection criteria, like the distance from the edge, and to then turn around and try to validate or use that to, you know, benchmark a predictive model. Would you reverse that weighting, or would you bake that weighting into the evaluation? Or I was wondering how if you've thought about that next step.
Yeah, this was so this weighting was kind of, I want to have at least variance for every position in my oligo that with a similar effect size. and wanted to learn something about it so that I don't select all variants that have high effects in the center, and at the end, on the edges, I don't have any high effect variants. So I want to have the distribution of all of it. I think it's better in terms of predicting later on. But yeah, I see also the challenge is that every of this MPRA might be done in a different system, and it's a different tissue, and it's hard to do this inter-tissue prediction activity. I hope that at least lots of us are going to use the same control set so that we get somehow variability. So between, okay, it can be between the groups that did the essay, between also maybe the tissues, and that this can be somehow put into the catalog or into the model later on.
I was thinking the same thing, Jesse, during Max's talk. Like, does he have to undo the sampling scheme? And like, for when they do a census, you sample hard to reach populations or populations with like higher variance. You prioritize places that are going to have high variance, so you have more samples there. And then you'd have to like undo that if you want to estimate something about the population. But I'm thinking like if the model that we're learning is general and doesn't have to do with the aspects of his sampling scheme, then you don't have to undo it, I think. So it depends on whether the properties that he uses to sample the variance, or like to reassign which variants go into the experiment. If those also correlate with the effect sizes, then you maybe have to be concerned about that. But if it's a general model you're learning, I think you don't have to worry about it.
Yeah. Then when I have to worry about it, then I generated a good benchmark data set and not a good training data set, because I have on every position different all range of effect sizes that then it could be difficult for training. Yeah, but this is steps beyond. First of all, I need the data to see at least what. Yeah. Yeah, but I have to consider this more carefully again when. Oh, thanks.
Yeah, thanks a lot. And I think there are two different issues. You pointed out that there are actually two different considerations to be thinking about here. One is about balancing in the selection of variants to test, and then the other is including the right controls so that we can actually compare values across different experiments and assays and cell types. And I was wondering, I guess, general question for everyone, if from the presentations, from the different groups that we heard today, do we feel like if we wanted to combine these 4 data sets, or I don't know, three specific ones that were proposed, and cross-analyze them and compare variant effects, would these designs enable that? I guess this is a question maybe first for the speakers.
So you're speaking just about the controls that are used, or about the whole data?
Yeah, I guess it could be controls and then also experimental standards. But I mean, just thinking about the control sequences, I really liked all of the controls that were described. And then there was some overlap between the groups, but not perfect overlap yet, that we can think about collecting all of these lists. But I was just wondering if as you were presenting or listening to the other talks, if there are things that you thought were clearly missing.
Yeah, I think for the controls, I think there are these scrambled sequences. These are, I think, in general, nice negative controls that should work in every assay. A lot of I think groups just select specific control sets on their, let's say, tissue of interest. And yeah, so then it just comes up. So if we just include, it doesn't include all controls from all MPRAs, but you will see natural differences here. And yeah, the question is, can we predefine some, this is positive in all and this is negative in all? I think that's a really difficult question to tell. And also it's, I think then try to test them on another set, or try to use the controls on different sets. And so what are the controls? I think we maybe can agree on a few smaller negative sets and maybe some positive control sets, but I think this is again a really difficult challenge. Maybe the first round of the design will brings us more closer to that. I'm not sure.
Another thing, Max, that I would like to add is even our group has designed positive controls largely based on the element level, while actually the entire goal of our MPRA teams are designing defining the variant effects, right? And we simply don't even have enough controlled data sets when it comes to the variant level. And I believe that this positive enhancer elements would be very useful to see whether there is this difference between negative versus positive controls that kind of gives an overall idea that the MPRA has run well or not. But I'm not sure whether that can be necessarily used as a benchmark to verify that these are the major factors to govern the variant effects.
Yeah, did anyone think about ChIP-seq, if they have ChIP-seq data, looking on the specific transcription factor and try to kind of disrupt it using, just looking at the motif and change the major variant, a major position here?
That's actually a great idea. I think Nick actually mentioned that he has subsetted some of the variants based on DNA's footprints. That could be something that is useful for that purpose.
Yeah, we'd love to do saturation mutagenesis of some of those footprints eventually, but I don't think it'll fit in our current library design. But I want to bring up another sort of idea that we've talked about in our focus group, which is designing synthetic sequences that don't occur naturally but are predicted to be expressed in every genome. For example, if there's a synthetic sequence that you just tile with a bunch of constitutive promoter sites, I mean, we would love to use something like that as our global control data set for MPRA, and I think that solves a lot of problems across these different experiment comparisons. I just don't think we have a good idea to start, and that we'd have to do a whole separate experiment to validate those synthetic controls, which pushes back all of these interesting biological questions by months to a year. But if there's anyone on the call who might be interested in helping with that problem, we'd love to chat.
Well, so we have I don't know the exact number, but we have a set from Qingyu of artificial sequences with transcription factor motifs inserted. So we should all include those. They're up on in our project design. I mean, they're from Qingyu, so you have them.
There's about 600, I think, ish, but they're all MPRA validated with RNA, DNA ratios are included in them. Nick, you're 270 base pair sequences, right?
Yep.
Yeah, so these are mostly 270 that we pared down to 250 for our design, but yeah.
And are those in
Thank you.
a particular cell type, or have they been tested in multiple?
I think just in organoids and primary cells.
What? Yes, HepG2 and in HNP.
Fetal cortical cells too, I think. There's some.
Yeah.
Yeah, but again, this is just regions of the. Again, it's not the kind of variants. Here we can somehow try to modify the inserted transcription factor to get variants. So this would be a control plus from those. Maybe that's something also that's maybe our start for getting positive or negative control variants.
So yeah, and there's also a good, there are like some positive control variants. So we've got like 4 bat types, John.
Yeah, let me pull it up. Yeah, there's the islet beta cell. Well, it's not strictly from Karen that have allelic difference.
But yeah, you want them to be general across.
Well, that's the problem, right? Yeah. I don't know how many we have that are validated, at least general across multiple.
And Max, if it was synthetic sequence, will you perturb every motif, or it'd be difficult?
Yeah, synthetic sequence. Yeah, that's why I thought about chipstick peaks. So where you're looking for this transcription factor, you see a peak in your specific cell type of interest. You know this motif, and you maybe can change important positions in the motif.
So I was just looking at something recently from Karen, where if you have a heterozygous site, and I have you can look at the allelic effect in the chip. It's very clear signal that there's a effect on binding, and maybe we could find a ChIP-seq peak that's kind of universal across encode cell types or something. Yeah,
that would be nice. Yes.
Maybe some CTCF motif, although I guess that probably its effect will vary depending on the vector format.
But CTCF. I don't call it transcription factor, but
another category I was wondering is whether it would make sense to put promoters of interferon responsive genes in all the libraries to sort of monitor this interferon transfection response that the STARR lab has documented. Or maybe if this list of synthetic sequences with transcription factor motifs added includes interferon as one of the motifs, that would be perfect.
Yeah, we've seen that in the already live in STARR-seq. I don't know if in MPRA there's the same. I guess it has to do with the transfection, and I guess that's pretty universal in many of these libraries. So I guess so, but that's a good point, Jesse.
So we're now 2 hours workshop. We can of course still continue discussion if we have time. Otherwise, we can continue discussion in the focus group. MPRA pipeline, again, not really designed. Maybe we should start a new focus group about MPRA design, but anyway, it's fine. No, not more focus group, not more calendar invites, not more meetings. I see. But I really like to thank everyone who's participating and all the speakers. I think, thank you. And we definitely do another workshop about later when we have the data about the analysis. But I think we definitely keep in touch about the design and controls, and we just continue, like in the Slack channel, the discussion. And let everyone think about what variant controls we gonna use. That would be great.
Yeah, and we should pull all together. I was just gonna say, discuss negative control coordination, because I feel like we've been focusing on positive controls and not so much negative controls. So,
All right.
Thank you, everyone.
Goodbye.
Great talks.